Assalamu alaikum everyone. My name is Baqir and I will be giving a presentation on the key paper review. So the paper that I chose for the key paper review is titled Cancer Cell Imaging and Photothermal Therapy in the Near Infrared Region using Gold Nanorods. So let's get started. Um, Before I begin the review of the paper, let's just discuss some important details about this paper. So this research article was published in the Journal of the American Chemical Society in the 2006 edition, and it has 5,341 citations as of now, according to Google Scholar, and this highlights how important and fundamental this research article is. And in fact, one of the key contributors and one of the key authors of this article, Mustafa Al Sayed, who is an Egyptian chemical physicist, is a leading nanoscience researcher and a member of the National Academy of Sciences and a U.S. National Medal of Science laureate. And also, he and his group have contributed to many important areas of physical and materials chemistry research. And his research includes the use of steady state and the ultra fast laser spectroscopy. And his group is also involved in the development of new techniques such as picosecond Raman spectroscopy and microwave double resonance spectroscopy. And his lab is also known for the development of the gold nanorod technology. And as of 2021, he has produced over 1,200 publications in refereed journals in the areas of spectroscopy and molecular dynamics, and which actually shows how important a researcher he has been. And talking about this very research article, it is one of the very first research papers to investigate the use of gold nanorods in cancer cell imaging and photothermal therapy in the near infrared region. So let me give you a little bit of background of this paper. Um so first of all, what's so interesting about gold nanoparticles? Well, so basically, in noble metals like gold and silver, the electrons in the conduction band undergo coherent collective oscillations, which greatly enhance the radiative properties of gold and silver when they interact with resonant electromagnetic fields. Now, this makes the absorption cross section of these nanoparticles orders of magnitude stronger than that of more strongly absorbing molecules, and the light scattering cross section orders of magnitude more than that of organic dyes. Um, so, these particles act as excellent sensors and novel contrast agents for optical detection due to their enhanced absorption and scattering. So, in addition, when it is realized that the strongly absorbed radiation is converted efficiently into heat on a picosecond time domain due to the electron phonon and the phonon processes, their potential use in photothermal therapy becomes obvious. Now, the scattering properties of the gold nanospheres have been used for cancer cell imaging and using the confocal microscopy and simple dark field microscopy. Recently, photothermal therapy using the absorption properties of antibody conjugated gold nanoshells and solid gold nanospheres has been demonstrated to selectively kill cancer cells, leaving the healthy cells unaffected. Now, if gold nanospheres are working so well, then why do we need gold nanorods in the first place? Well, it turns out in order to increase the penetration of laser through several layers of tissues, we need a light of longer wavelength, somewhere in the near infrared region. So it is desirable to use agents that are active in the near infrared region of the radiation spectrum to minimize the light extinction by intrinsic chromophores in the native tissues. And gold nanorods with suitable aspect ratios can actually absorb and scatter strongly in the NIR region, which is about 650 to 900 nanometers on the radiation spectrum. Now, Now, the absorption band of these noble metallic nanoparticles is actually tunable by altering the nanoparticle shape or size. Um, so, other shapes of gold nanoparticles, such as branched pentagon and large prisms, have the surface plasmon absorption in the NIR region. But nanorods are better candidates for their application due to the ability to accurately control the absorption maximum to the required wavelength by changing the aspect ratio. And we can change the aspect ratio of these nano rods by simply changing the silver ion concentration during the gold nanorod growth process. So this is a graph which basically shows the absorption spectrum of gold of different gold nanoparticles at different wavelengths. So as you can see that the gold nanospheres have a very good absorption at around 500 to 600 nanometers, but that falls out gradually, very rapidly when the wavelength is increased beyond 500 nanometers. And whereas gold nanorods uh, have very high absorptions at even longer wavelengths. So now in this research article, um, the researchers provide an in vitro demonstration of gold nanorods as novel contrast agents for both molecular imaging and photothermal cancer therapy. So nanorods are synthesized and conjugated to the anti-epidermal growth factor receptor, monoclonal antibodies, and incubated in cell cultures with a non-malignant epithelial cell line and two malignant oral epithelial cell lines. And so the anti-EFGR antibody conjugated nanorods bind specifically to the surface of the malignant cells with a much higher affinity due to the overexpressed EGFR on the cytoplasmic membrane of the malignant cells. And the strongly scattered red light from the gold nano rods in the dark field uh, shows that the malignant cells are clearly visualized and diagnosed from the non-malignant cells. So it is found that after the exposure to continuous red laser at 800 nanometers, malignant cells require about half of the laser energy to be photothermally destroyed than the non-malignant cells. So both efficient cancer cell diagnosis and selective photothermal therapy are realized at the same time. So here's an image which shows a comparison between the scattering images of the anti-EFGR antibody conjugated gold nanospheres and gold nanorods. Now, the nanospheres. So the the A row shows basically the scattering images from using the gold nanorods, and the B images show the scattering images using the gold. Sorry, the A images shows scattering images using the gold nano spheres, and B images show the scattering images using the gold nano rods. Now, the nanospheres scatter, in green to yellow, from green to yellow light, because the surface plasmon oscillation frequency is in the visible region. And whereas the light scattering images from the antibody conjugated gold nanorods actually show light scattering from orange to red due to the strong longitudinal surface plasmon oscillation with a frequency in the NIR region. And now, these images show the effect of a laser beam, which is incident on different types of cells: the Hackett non-malignant cells, and the HSC malignant cells, and the HOC malignant cells at different intensities, at the different intensities. And the blue circles and the blue dots in between show the dead cells, and the circles basically show the laser spots on the samples. Now, at when we apply an 80 millivolts intense incident light, the malignant cells are uh, injured while the non-malignant cells are not affected. And well, when we increase the intensity of the incident radiation, the non-malignant cells also get affected. Now, coming to the key findings of this paper, so this study basically demonstrates that the gold nanorods are um, a novel class of optically active dual imaging therapy agents due to their strong absorption and scattering of the near infrared light. And by using the surface plasmon resonant absorption spectroscopy and light scattering imaging, the malignant cells are easily distinguished from the non-malignant cells due to the molecular targeting of overexpressed EGFR on the antibody conjugated gold nanorods. Uh, and so Gold nanorods are basically biocompatible, optically active absorber and scatterer, and that may potentially serve as the backbone for an assortment of designer compounds using a variety of vectors for molecularly targeted photodiagnostics and therapy. Let's come towards the shortcomings of the paper. Well, they're not exactly shortcomings, but something that has not been mentioned or taken care of in this paper is that how um, well, we have studied that uh, we can change the absorption range and the absorption band of the gold nanorods by adjusting their aspect ratio. But this paper doesn't really go into the detail when it comes to the binding affinity of these nanorods of different aspect ratios, um, and and their caping abilities with the antibodies. These things haven't been studied much in detail in this paper. And coming towards the state of the art, gold nanorods are obviously very promising metals in several biomedical applications such as bioimaging, thermal therapy, and drug delivery. And so the reason is that gold nanorods have strong absorption bands in near infrared region, and they also show photothermal effects. And since NIR light can penetrate deeply into the tissues, their unique optical, chemical, and biological properties have attracted considerable clinical interest. And interestingly, the Chemical Reviews journal actually uh, dedicated their 2008 edition towards gold, and there were dozens of articles on the use of gold nanoparticles in different areas of interest, and there have been a number of key papers uh, in this area which discover the different applications of gold nanoparticles for cancer detection and the photothermal therapy. That was it from my side, and thank you very much.